All right, now in Chapter 3, what we're going to do is take some of what we talked about in Chapter 2 and kind of build on it. And so one of the things we'll certainly be doing is we're going to be talking a little bit about organic compounds that are really important to living things. And so therefore we're going to be talking about carbohydrates. We'll also talk about lipids. We're going to talk about proteins, and we'll finish off. Now, one of the things we want to keep in mind is that these compounds we're going to be talking about, they're carbon-based compounds. So oftentimes we just refer to them as being organic compounds. So carbon-based compounds are organic compounds. Now, one of the questions we can ask, and by the way, we're carbon-based life forms. So, if we're carbon-based life forms and we go out on a daily basis and our food that we take in are carbon-based compounds, how does carbon really kind of get circulated throughout all living things, on the planet? Well, photosynthesizers are really responsible for getting carbon into forms that living organisms can use. Photosynthesizers take in carbon dioxide and they convert the carbon dioxide into carbon-based compounds of life. And this includes carbohydrates, proteins, nucleic acids, and lipids. If you remember, carbon can form up to 4 bonds. That's what makes carbon such a versatile element, because it has 4 unpaired electrons in its second energy shell. Now, we have a whole chemistry dedicated. It's a sophomore level chemistry dedicated to the study of carbon-based compounds. It's called organic chemistry. And this is because carbon is so important to our lives in so many different ways. Our economy is a carbon-based economy. I mean, the fuels that we use in our vehicles to get us to and from our destinations, these are carbon-based compounds. Our plastics that are so important to us in our lives are carbon-based compounds. Our clothing, if we have like cotton clothing as an example, that cotton is a carbon-based compound. Now, carbon typically forms diverse molecules by bonding to other atoms. And one of the atoms that carbon tends to bond to would be hydrogen. Oxygen is another common partner with carbon, as is nitrogen. So, once again, we can remind ourselves of the electron configuration of carbon. Carbon has a total of 6 electrons. It has 2 electrons in its first energy shell. It has 4 electrons in its second energy shell. And because of that, because it has 4 electrons in its second shell, and we can see that down here, here's 1, 2, 3, and 4, it tends to form 4 bonds. And so oftentimes we say carbon has a valence of 4. It can form 4 bonds. And so oftentimes we'll even use the term tetravalent in referring to carbon's ability to form 4 bonds. So down below we see this diagram again listing the big 4 that make up 96% of living matter. These are the elements that make up the bulk of living organisms, and they are frequent partners of carbon. Now, there can also be an awful lot of diversity to the molecules that carbon can be a part of. Some of these molecules you can see are going to be straight, like we have here. Some of them will actually be branched, like we see here with isobutane. And sometimes these compounds, these carbon-based compounds, will actually form ring structures. So there's a lot of diversity that can come out of these carbon-based compounds. And also, not only can they be straight, branched, or in rings, but the lengths of the carbon-based compounds can vary. Here we see it's a 2 carbon atoms to the skeleton. Here we have 3. Here we have 4, and so on. So once again, these carbon-based compounds, one will find a lot of diversity associated with them. Now, here's a term we'll use from time to time. We'll talk about hydrocarbons. Now, hydrocarbons are compounds, like we have over here, that consist of only hydrogen and carbon. Now, if you guys remember back in Chapter 2, we noted that This bond between a carbon and a hydrogen is a non-polar covalent bond because the electronegativities of carbon and hydrogen are pretty similar. And so, remember, this line represents a shared pair of electrons between the hydrogen and carbon, and they pretty much share the pair of electrons equally because their electronegativities are very similar. Hydrocarbons consist of only hydrogen and carbon. Our fossil fuels that are so important in generating electricity for us and getting our vehicle down the road are hydrocarbons. Because of the nonpolar covalent bonds between carbon and hydrogen in the hydrocarbons, then these compounds will be hydrophobic, which means, once again, hydro referring to water, phobic referring to fear. These compounds that are hydrocarbons, they're hydrophobic. They do not mix well with water because of their relatively non-polar covalent bonds they have between the carbons and the hydrogens. But the thing about these hydrocarbons is that they really are a good source of stored energy. I mean, think about the gasoline you put in your vehicle. It's a very good source of stored or potential energy. Something else that we have with some compounds is that some compounds, we can have two different compounds. They each have a different name. Like here we have butane and isobutane. But you know what? When we look at them more closely, we're going to see some real similarities. The obvious difference is their shape. But what's similar about them? Well, butane has 4 carbons plus 4, 8, 9, 10 hydrogens. What about isobutane? Well, it too is made up of 4 carbons and 3, 6, 7, 8, 9, 10 hydrogens as well. So both butane and isobutane have the same number of carbons and hydrogens. So they're the same in that way, but their shapes are different. So we refer to butane and isobutane as being isomers. Isomers are compounds with the same number of atoms of the same elements, but they differ structurally from one another. Remember what we've said before. Shape dictates function. So even though these two compounds, they're the same in terms of number of carbons and hydrogens, their shapes are different, so they're going to have different properties. Now, another kind of isomer grouping that we find is differing placements of double bonds. Here we have a double bond in 1-butene coming off the number 1 carbon. That's why it's called 1-butene. And by the way, ene tells you there's a double bond. 1 says it comes off the number 1 carbon. Over here we have 2-butene. Ene, double bond coming off the number 2 carbon. So the placement of the double bond varies between these two compounds. Other than that, they're the same. Essentially. I mean, placement of hydrogen varies a little bit. But they have the same number of carbons, same number of hydrogens. Placement of the double bond is different. So these two compounds are isomers. Now, isomers matter. For instance, we have some isomers that bend polarized light to the left, so we call those levorotary compounds. We have some that bend polarized light to the right. We call those dextrorotary compounds. And the interesting thing is, we may take, say, ibuprofen to help when we have experiencing pain or inflammation, but it's the left-handed form of ibuprofen that actually will help us with pain relief and deal with inflammation. The right-handed form of ibuprofen will not help us with pain and inflammation. Now, one of the things that's kind of interesting, though, is that within our cells, we have an enzyme that can convert right-handed ibuprofen into left. But these two compounds, one of them bends light to the left, one bends light to the right, but they have the same number of the same kinds of atoms. They are isomers, but one isomer works for pain and inflammation, and the other one doesn't. Albuterol is another example. There's right-handed butyrol, which is the isomer form that works for asthma, and there's the left-handed form that does not. So isomers, they're going to have the same number and kinds of atoms, but they're going to have some differences in their structures, in their shapes, and so they will have different properties as well. Now, what we're going to do is we're going to work our way through some chemical groups. Chemical groups are clusters of atoms that tend to occur together, and they bring with them, or they help create certain properties of molecules, and also they oftentimes will participate in chemical reactions. Now here we have a male line and a female line, very obvious to us, and here we have a peacock, which is male, and a peahen, which is female. Now down here we have drawings of two very important molecules that help, shape males in a male way, help shape females in a female way, whether we're talking about lines or whether we're talking about peacocks, peahens. Now, if we compare testosterone to estradiol, we're going to see lots of similarities. We have one ring, two rings, three. Here's a fourth ring. We have one ring, two, three, four rings for estradiol. So testosterone and estradiol both are composed of a four-ring base. Also, coming off of this first ring, there's an OH group. This is one of the chemical groups that we're going to be looking at. This OH group is a polar group. The oxygen has a partial negative, the hydrogen has a partial positive, and that really helps makes the estradiol a bit more water soluble. Because guys, these rings are actually representing a bunch of carbons and hydrogens, but the OH with the partial negative on the oxygen, partial positive on the hydrogen, makes, or at least this part of the compound, hydrophilic. This is one of the chemical groups we're going to be looking at. Well, testosterone has that same chemical group in the exact same location. Here's a methyl group. This is another chemical group we're going to be looking at. Testosterone has that same chemical group. But look, here's the difference. Testosterone has a methyl group here, estradiol doesn't. Estradiol has a hydroxyl group here, testosterone doesn't. So we find just a few differences between testosterone and estradiol in terms of chemical groups. Other than that, they're pretty much the same. But this guy produces larger quantities of testosterone than the female line. But she's producing more estradiol, certainly, than the male. The peacock's producing more testosterone, the female's producing more estradiol. And so it's interesting when you look at testosterone and estradiol, the compounds look very similar, but the effects they have on the individuals producing them is dramatic. Okay, so now what we're going to do is go ahead and take a look at chemical groups that are associated with biologically important molecules. So we're going to keep in mind that these chemical groups are going to have an effect on the shape of the molecules to which they're attached, and oftentimes they will improve the solubility of the molecule in water. And of course, when we think of improving solubility in water, and we're talking about living organisms, that's certainly critical, because the largest percentage of an organism is really water weight. Now, the first five that we have listed over here in Table 3.2 are called functional groups. So the hydroxyl group, the carbonyl group, the carboxyl, the amino group, and the phosphate group. These are all functional groups. Now, we separate those five out and call them functional groups because oftentimes they're participating in chemical reactions within cells. Now, a lot of these groups we're going to find anyway, have some polarity associated with them. And what that does, again, is by there being some partial charge associated with these functional groups, that will make the compound to which they're attached more hydrophilic, which means it's more easily transported in the blood or in and around cells. So the water solubility improves with the attachment of a functional group. Now, the first functional group we're going to look at is the hydroxyl. The hydroxyl is an OH group, one of the things we want to make note of about the hydroxyl is that there is a polar bond between the oxygen and the hydrogen. Remember, there's a shared pair of electrons between the O and the H, but the electronegativity of oxygen is much greater than that of hydrogen. So the shared pair of electrons between the O and the H spend more time around the oxygen nucleus, less time around the hydrogen nucleus, giving the oxygen a partial negative charge, the hydrogen a partial positive charge. So that's something we want to keep in mind. Now, when we look at the carbonyl group, we're going to see that it's a carbon with a double bond to an oxygen. Now, sometimes the carbonyl is part of what we call a terminal carbon, a carbon that's on the end of the molecule, like we see here. Anytime we have a terminal carbonyl, we're going to call it specifically an aldehyde. If we have a carbonyl that is not part of a terminal carbon, like we have here, then we're going to call it specifically a ketone. So aldehydes and ketones are different kinds of carbonyls. They're both carbon double bond to oxygen, but one involves a terminal carbon, the other one's a non-terminal carbon. Carbonyls. Aldehyde if they're on the end, if they're a part of an end carbon or terminal carbon. Ketone if it's a non-terminal carbon. Carboxyl is another functional group, and when you look at a carboxyl, you'll see it's a carbon with a double bond to an oxygen and a single bond to an OH. Now, when you look at this, it seems like what we have is a carbonyl here and a hydroxyl there. But the thing we can't do is we can't break up carboxyls into carbonyls and hydroxyls. So when it looks like we have a carbonyl with a hydroxyl, then that in fact is a carboxyl group. Now, here's one of the things that's unique about carboxyls that we'll want to remember, is that carboxyls can lose this hydrogen as a proton, which means the hydrogen leaves its electron behind with the oxygen to which it was attached, and it can come off as a hydrogen ion. So that means this group can donate hydrogen ions to its environment. And so because of that, we're going to remember that carboxyl groups behave as acids by donating hydrogen ions to their environments. The next functional group is the amino group, which is an NH2 group usually. Here we have an amino group NH2. You may remember that a nitrogen atom has 3 unpaired electrons in its valence shell. It also has what's called a lone pair of electrons in its valence shell as well. And that lone pair of electrons can take up a proton from the environment. Now remember, anything that can take up a hydrogen ion, a proton, is a base. So amino groups can behave as bases because they can accept. So here's an amino group before, and here's an amino group after it is taken up, it is accepted a hydrogen ion. Phosphate groups, a very important functional group. The phosphate group carries negative charge, and the phosphate group is an integral component of adenosine triphosphate, ATP. And that's what this molecule is. It's ATP, adenosine triphosphate. Now you see there's negative charge associated with some of the oxygens. Now, one of the things that we'll kind of keep in mind is that these 3 negatively charged phosphate groups clustered together, is really a system of instability. But we will want to keep in mind that when something is relatively unstable, it's typically a good source of energy. Think of gasoline. Well, the last functional group that the last chemical group, it's not a functional group, but the last chemical group we're going to look at is the methyl. The methyl is a CH3. That's the methyl group, and one of the things that they're finding as they study human chromosomes and gene expression is that oftentimes genes that have been turned off or silenced have a lot of methylation. They have a lot of methyl groups attached to them, which they seem to prevent appropriate cellular enzymes from getting to the gene and getting the gene expressed. So methyl groups are important in that way. Now what we're going to do is we're going to now look at biologically important molecules. So in Chapter 2 we did some basic chemistry stuff. Here in the beginning of Chapter 3 we've been looking at functional groups. Now we're going to talk about molecules that are really important to living things. And there are 4 that are really important to us. And you're familiar with these: carbohydrates, proteins, nucleic acids, and lipids. We're going to talk about these 4 biologically important molecules. We're going to start off talking about carbohydrates. Now, when we start talking about carbohydrates are polymers. Poly means many. So over here we have a short, generalized drawing, a short polymer. And what a polymer consists of are monomers. So this purple sphere represents a monomer, a single unit. But it's bonded to another single unit, another monomer, which is bonded to a third monomer. But collectively, since they're all bonded together, we call it a polymer. Many subunits bonded to one another. And here we have a reaction in which this monomer is about to be added to this one to create a 4-unit polymer. Now, the kind of reaction that will be carried out to attach this monomer to this growing polymer is called a dehydration reaction. It's called a dehydration reaction because a hydroxyl off of the growing polymer and a hydrogen off of the unlinked monomer will be joined together, forming water. So an enzyme will pull this hydrogen, this OH off, combine them in, form water. Water comes out as the bond forms. As this bond forms, water has left. Water is gone now. And so it's called a dehydration reaction for that reason. So it's a dehydration reaction that forms the new bond as a polymer is constructed. And carbohydrates are polymers. Now the monomer of a carbohydrate, in other words, like a single unit of, say, a carbohydrate, will be a monosaccharide. So this would be one monosaccharide bonded to a second monosaccharide, bonded to a third saccharide, and so on. Glucose, fructose, these are examples of monosaccharides. Proteins are also polymers. Now, in the case of a protein, though, the monomer is going to be an amino acid. Now, there are 20 important amino acids in proteins that make up proteins that are important to humans. So this would be one amino acid. This could be a bonded to a second amino acid, which is bonded to a third amino acid. And the way a fourth amino acid is hooked to this growing polymer of amino acids is once again by way of a dehydration reaction. An enzyme pulls a hydrogen off of an amino acid, and then hydroxyl off of the growing amino acid chain. Once again, it's put together to form water. Water comes out as the bond forms. So now we have 4 amino acids hooked together. We're building a large protein. There are, once again, 20 different amino acids that are monomers that are used in the building of proteins. Now, a third kind of polymer are the nucleic acids. This is like our DNA and our RNA. Now, the monomer would be a nucleotide. Now, in the case of DNA, you may be familiar with this. DNA nucleotides will be the A-type nucleotides, the T-type nucleotides, the G-type nucleotides, the C-type nucleotides. So you've probably heard of A's, T's, G's, and C's associated with DNA. So what we have in the case of nucleic acids is we have one nucleotide bonded to a second, bonded to a third, and so on, and that makes up our DNA, okay, and also our RNA. We'll look at that a little bit later. So there are 4 different nucleotides, A's, T's, G's, and C's. Now, the fourth biologically important molecule will be the lipid. But lipids are not polymers. They're not constructed from many monomers hooked together, okay, and they're not even a macromolecule. They're not really all that big usually. So the top 3 are polymers constructed from monomers, and they're macromolecules. They tend to be pretty large molecules. Lipids, in relative terms, tend to be smaller, so they're not macromolecules. They're not constructed of hooked together monomers. They're not polymers. So from the previous slide, the first 3 are macromolecules. And we've noted that cells can link monomers together by way of dehydration reactions, and that's because water is removed as the bond forms. And once again, we illustrated that back on the previous slide. Now, think about this. Here's a question we have. How can there be so many different proteins being produced by cells? Because we make thousands of different kinds of proteins, and yet they're all constructed from the same 20 amino acids. How can there be so much diversity in our DNA when there's only 4 different nucleotides, A's, T's, G's, and C's? Well, an analogy might help us with this. You know, there are only 26 letters in the American alphabet, but think of all the different words that are constructed from the 26 letters based upon different letter sequences, the number of letters within a word, and so on. And that's kind of how we can get different proteins, different amino acids in different sequences of different lengths, and same with nucleic acids as well. Now, once we take in food, we're taking in polymers. Think of the carbohydrates that are listed in grams on your food labels. Think of the grams of protein that's listed on that food label. These are polymers we're taking in with our food that are actually food polymers. Now, what we're going to do during digestion is we're going to break down the polymers back into monomers. Now, the way we're going to do this, if you remember, when the polymers were constructed, water came out in a dehydration reaction. So part of breaking the polymers down into monomers will be the adding back of water, and we call that hydrolysis. So here we have this polymer. We need to break this bond. So we're going to have an enzyme that's going to help. That's not being shown here. But we're also going to have the addition of water, so we can get a hydrogen from the water to go to a monomer that's coming off, and then the remaining OHs be added to the end of the polymer chain. So that's what we're doing during digestion, breaking the polymers of food down into monomers by way of hydrolysis. The word hydrolysis really means using water to break, and we're breaking the bonds between the monomers. And once again, we will talk more about enzymes and how they help a little bit later. Now, enzymes are critical. They're really important to us. They are really kind of workers of cells. What's interesting, though, is that we don't always produce enzymes every single day, 24 hours a day, 7 days a week, 52 weeks a year. We produce them when we need them, usually. Now, one of the things that's interesting, though, is that some individuals in their lifetime, really after about 2 years of life, after they're a 2-year-old or so, on average, can sometimes develop something called lactose intolerance. Well, this is because they no longer produce the enzyme lactase. Now, they have the ability to produce it, they just don't. So they are no longer producing the lactase enzyme. So they cannot break down, they cannot hydrolyze lactose. Now, almost all infants can break down lactose. That's milk sugar. And it's really interesting that for many people, they turn off that gene producing lactase after about the age of 2. Now, this is what's pretty normal, actually. As a matter of fact, about 80% of African Americans and Native Americans stop producing lactase after the age of 2. Or at least there's a significant decline. There's the beginning of the decline in producing lactase after the age of 2. And so oftentimes, by the time these individuals are in their teens, they are not only lactase deficient, but they're lactose intolerant. Now, what's interesting about Americans of Northern European descent is that they continue, many of them anyway, continue producing lactase even into adulthood. Now, researchers have looked into this, and one of the things that they think is probably going on is that the story they've been able to put together anyway is that Europe's cold climate, which would allow only about one harvest per year, favored those ancestors who had a mutation in their lactase gene that kept the gene turned on throughout life. Now, what that meant then is that these individuals, they could still get energy and they could get their nutrients from milk and other dairy products year round, because they certainly weren't going to be able to grow crops or many different crops anyway during the cold times of the year in those colder climates. So what they would do is they would use their domesticated cattle, which they've had for 9,000 years or so, to really supply them with both energy and nutrients, and they could handle the lactose because they had a mutation in a gene that allowed them to do so. So they would remain healthy and still be able to add to their families in terms of number of children, They've also found the same kind of thing going on within many East African groups that are reliant upon dairy herds. These individuals also are lactose tolerant. Now, what they've found with these individuals is that they have 3 different mutations. And these mutations are different from what they find in the European mutation. So it's kind of interesting that, different ways in different peoples and different geographic regions in terms of, having this lactase gene remain on longer so that they can still, be lactose tolerant and derive energy and nutrients from dairy products. Well, let's work our way through these biologically important molecules. We're going to start off with carbohydrates, specifically monosaccharides. We have 2 examples of monosaccharides. We have glucose and we have fructose. Now notice it reads glucose is an aldose. That's because this functional group, which you should recognize as a carbonyl, and since it's a terminal carbon that makes up the carbonyl, it would be an aldehyde. And sugars are referred to as oses oftentimes. So since it's an aldehyde sugar, it's an aldose. Fructose, on the other hand, is a ketose because that really is a ketone. And ose for sugar. So sugar names, monosaccharide names, oftentimes end in ose. But monosaccharides are simple sugars. They are the monomers of carbohydrates. Now, honeybees, it's kind of interesting. They make up both glucose and fructose when they make their honey. So honey consists of glucose and fructose. Now, if you look at the term carbohydrate, carbo might remind you of the word carbon. Hydrate, maybe water. Think of hydrating yourself with water. And when you look at the empirical formula of carbohydrate monosaccharides, what do you see? Carbon and what? Water, H2O. So the proportion of carbon to hydrogen to oxygen, it's a 1-to-1 ratio. That's the empirical formula with monosaccharides. Now, here it wants us to identify the trademark functional groups associated with monosaccharides. Well, we've already identified what? Carbonyl, that's common. And what else? OHs, there's one OH, second, third, fourth, and so on. Multiple hydroxyl groups. That's common in sugars. And we're going to keep in mind these hydroxyls are polar. And so that's the solubility of these monosaccharides. Now, here's another question. Do glucose and fructose, this reminds us of some other material we looked at a little bit ago, do glucose and fructose have the same number of carbons, hydrogens, and oxygens? And you know what? You're going to find that indeed they do. Now, since they have the same number of carbons, hydrogens, and oxygens, but there's some difference in their shape, what are these 2 compounds then? They are isomers of one another. So that's the term that applies to this. They are isomers. Now, would you expect glucose and fructose to have the same properties? No, because they have what? Different shapes. Exactly. As a matter of fact, fructose tastes sweeter than glucose because of its shape being different from glucose. Now, there's been a lot of talk, over recent years about high fructose corn syrup. So what is high fructose corn syrup? We can see down here in this picture of Coca-Cola, one of the main ingredients is high fructose corn syrup. So what is high fructose corn syrup? Well, it's where cornstarch is industrially hydrolyzed into glucose. That makes corn syrup. So cornstarch is hydrolyzed, which means what? The starch, which is a carbohydrate, it's a polymer, is hydrolyzed into monomers, into the glucose monomers, and that is corn syrup. Then enzymes are used to convert these glucose monomers into fructose, into the isomer fructose. Then the fructose is combined with some of the corn syrup to make the high fructose corn syrup. That's what high fructose corn syrup is. It's about 55% fructose and 45% glucose. So This is really not much different from our table sugar, which is sucrose. It's pretty much about the same ratio. So what makes glucose and aldose and a fructose a ketose? We've already looked at that on the previous slide, so I'm going to move on from there. I've also mentioned that most sugar names will end in ose, and most enzyme names, you've probably picked up on this already, end in ase. So, for instance, lactose is the sugar upon which lactase can act upon or can hydrolyze. Now, monosaccharides are really important to us because they are the main fuel for our cells. Once we get into talking about cellular respiration, production of ATP, we're going to see that glucose specifically is a monosaccharide that our cells love to use to make ATP. So glucose is especially important to us. Now, if you look down here, what we have is we're going to take glucose, and we see it's in a ring form. Why is it drawn up in ring form? It's drawn up in ring form because glucose does not remain linear in aqueous environments, like inside our blood or inside interstitial fluid or something like that. So in aqueous solutions, most 5 and 6 carbon sugars, and glucose is a 6 carbon sugar, as is fructose, convert into ring form. Okay? Now, do you remember we talked about the empirical formula for monosaccharides being CH2O a bit ago? What would be the formula for a 3-carbon monosaccharide bearing that 1:1 ratio in mind? If it's a 3-carbon monosaccharide, it would be C3H6O3. What would be the formula for a 5-carbon monosaccharide? C5H10O5. Well, that's a quick look at monosaccharides. Now we're going to move up to disaccharides. This is when we have 2 monosaccharides that have been joined together by a bond. It's actually called a linkage. It's actually called a glycosidic linkage. And once again, it's an OH off of one monosaccharide and H off of the other being put together, and since that forms water, water comes out as the linkage forms. We call it a dehydration reaction. So 2 monosaccharides can be joined by a dehydration reaction. And as a matter of fact, a disaccharide that we're all familiar with is sucrose. It's table sugar. It's primarily derived from sugar cane and sugar beets, but a couple others would involve lactose, which is milk sugar, and maltose, which is used like in the brewing of beer. Well, besides monosaccharides and disaccharides, there are also polysaccharides. Now, this is where we're going to get into looking at hundreds and literally thousands of monosaccharides that have been joined together by dehydration reactions. Now, a good example of a polysaccharide is starch. So, if we look down here, if we were to take a close look at a potato tuber using a microscope, what we have here is this is the cell wall of a plant cell. Inside here, these are starch granules. And these starch granules are just that. There are a lot of starch molecules making up the granule. This is a glucose monomer. Here's a glycosidic linkage that was formed by dehydration reaction with this glucose monomer. Glycosidic linkage to this glucose monomer, and so on. So what we see here making up starch is we see that there's just a lot of glucoses hooked together. And starch is the storage polysaccharide form for plants. Now, why do potato plants form tubers loaded with starch? Because what's going to happen is after the growing season and as winter comes on, the shoot system, the above ground part of the potato plant, will die back. And then next spring, all that energy that's stored in the tuber will be used to generate a new shoot system. So, the potato plant's not loading up with starch in its tubers for us, but rather it's setting itself up to grow a new shoot system in the next year's growing season. Once again, starch consists of glucose monomers, and here's something else. There's very little branching associated with starch. Okay, and there are a couple of different kinds of starch. Well, another kind of polysaccharide is glycogen. Now, animals like ourselves do not store our polysaccharides as starch, but rather glycogen. We store our glycogen in our muscles. We store our glycogen in our liver. And look at what glycogen consists of. Glucose, glycosidic linkage because of dehydration to another glucose, glycosidic linkage to another glucose, and so on. So what is glycogen? It's a bunch of glucoses hooked together. Glucose is the monomer, just the same as it was in starch. So what's the difference between glycogen and starch if they're both made of glucose monomers? Well, look at the branching in glycogen. You don't see that in starch. So that's the key distinction, is the amount of branching that you find in glycogen. It's highly branched. Another kind of polysaccharide has to do with, up here we can see the cell wall around this plant cell. If you look closely at that cell wall, you would see it's made up of glucoses hooked together in very straight strands that are hydrogen bonded to one another. And each of these strands is a cellulose molecule. Cellulose is the structural polysaccharide of plants, specifically their cell walls. So it's integral to the integrity of cell walls of plant cells. And I've already pointed out that the hydrogen bonding does add a lot of strength to plant cell walls. So think of something like wood, like a 2x4 or something. That is cellulose, and there's a lot of strength to it. We build our homes out of it. Now, humans cannot digest cellulose. We refer to it as being fiber or roughage in our diet. But there are some organisms, we think of like termites, for instance, being able to digest wood, but it's really not the termite itself that's digesting the wood. Really, in the gut of the termite lives this little teardrop-shaped protozoan called trichonympha. That's one species anyway, and inside the trichonympha are bacteria. So the termite ingests the wood, which makes its way there in the gut where the trichonympha and the bacteria are, and it's the bacteria that produce the appropriate enzymes for digesting, or hydrolyzing is the appropriate term, hydrolyzing the cellulose into individual glucose monomers. And there's also another kind of carbohydrate. It's called chitin. In the summertime in Texas, you'll hear this loud insect noise coming out of the trees around your house, around your home in the heat of the day. Well, those are cicadas. And you'll see the exoskeletons of cicadas on your trees. So it's kind of that brown insect casing that's on your trees. Well, that insect casing, that exoskeleton, has a lot of chitin in it, and it makes that exoskeleton tough so that they're afforded some protection. Well, chitin is another structural polysaccharide that insects use. And also, if you've ever handled certain kinds of fungi, you'll notice that the fungi is pretty tough stuff. I mean, not all mushrooms are soft and gooey. Some of them are really crusty, and it's the chitin that makes the fungus so tough. Well, another biologically important molecule or set of molecules will be the lipids. The thing about the lipids, though, is that they're pretty much hydrophobic, and you should be able to tell why. Look what they're made of: carbons and hydrogens. And what do we know about the bonds between carbons and hydrogens? They're pretty much nonpolar bonds, equal sharing of electrons, so no charge, because the electronegativity of a carbon and a hydrogen is pretty similar. Well, when you look at, we're going to look at fats, and then we'll talk about some other types of lipids as well. But when you look at something like a fat, one component you're going to see is you're going to see the glycerol here. This is one part of a fat. It's a glycerol. It's a 3-carbon compound. Here's 1 carbon, 2, 3. Okay? Now these oxygens coming off of each carbon are the remnants of a hydroxyl. Now remember what we said. Hydroxyls are functional groups, oftentimes participating in chemical reactions. So the glycerol is a 3-carbon component with an attached hydroxyl. You don't see the H with the O because it was part of a dehydration reaction, or lost in a dehydration reaction is what I should say. Now, another part of a fat is the fatty acid. So this hydrocarbon component is a fatty acid. Now look at that fatty acid. Now, which functional group behaves as an acid? It's a carboxyl. This COO is not a carbonyl. Instead, it's the remnant of a carboxyl. Here's a remnant of a carboxyl here and here. Once again, carboxyl and hydroxyls are functional groups participating in chemical reactions, and sure enough, they reacted with one another to form the bond here. Okay? This is one fatty acid, this is a second fatty acid, this is a third. Now, since there are 3 fatty acids, we call this compound, this whole compound, we're going to call it a triglyceride. So fatty acids have a nonpolar hydrocarbon chain with a carboxyl on one end, up the glycerol end. The nonpolar hydrocarbon part is hydrophobic. Fatty acids and glycerols are joined by way of dehydration reactions between the carboxyl and the hydroxyl in a dehydration reaction. Triglyceride is what we're seeing here in our drawing. So oftentimes we just call them fats, but they're really many times triglycerides. Now, we think of fats as just being bad, but they're not. It's like too much of anything can be bad, it seems like. Well, because, fats, they're important to us as long-term energy storage. I mean, 1 gram of fat has twice the energy of 1 gram of carbohydrate or 1 gram of protein. So, they're very important to us in terms of long-term energy storage. And there's not as much water being stored with fat cells. And so fat tends to be a little bit lighter in weight. And it's very energy dense. So that's great for mobile organisms like animals, like ourselves. We can carry around a lot of energy and it not weigh a lot. We also use fat to cushion our vital organs and to provide insulation. So, fats are not all bad. It's just too much can be a risk factor. Now we're going to break fats down into 2 groups, saturated fats and unsaturated. When you look at this fatty acid chain right here, you notice that between the carbons, from carbon to carbon, it's a single bond, single bond, single bond, single bond. From carbon to carbon, we see only single bonds. So this fatty acid is a saturated fatty acid because it has only single bonds. There are no double bonds in this hydrocarbon component of the fatty acid. Now, that's what we have here as well. Now, since there's only single bonds, that means you can have 2 hydrogens attached, except for the final carbon. But you have a maximum. You have the maximal number of hydrogens attached, which is 2. But look what we have over here. Single bond here, single bond, single, 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 but double. When you have a double bond between 2 carbons in the fatty acid chain, look what you can only have. Only 1 hydrogen attached to each of the 2 carbons that's involved in the double bond. That's not saturated, right? Double bonds do not allow a maximal number of hydrogens to attach to carbons. Okay? That would be 2 and 2. This is 1 and 1. So what we have in saturated fats, and by the way, this fatty acid is saturated, this fatty acid, but this is an unsaturated fatty acid. So overall, the fat here would be an unsaturated fat. So saturated fats will have carbons that have a maximal number of hydrogens attached. And also, you think of like butter and lard or something. Those saturated fats, like butter, will remain solid at room temperature. That's common in saturated fats. Think about that fat around the edge of a steak. It's solid at room temperature. Most animal fats are saturated fats. Now, on the other hand, in unsaturated fats, remember what we've said. You're going to find double bonds in the hydrocarbon chain. 1 double bond, like we have over here in this fatty acid, means this fatty acid, in this whole fat actually, there's 1 double bond. We would call this fat a monounsaturated fat. Now, if there are many double bonds in the fatty acid chain, so we have one here, let's pretend we had a couple here and a couple in this fatty acid chain. There are many double bonds. We would call it a polyunsaturated fat. And remember, where we have double bonds, we have less than the maximal number of hydrogens attached. And unsaturated fats will be liquid at room temperature, so we call them oils. So they will be liquid at room temperature, not solid. Now, the reason for this is because cis double bonds, which is what you have here, cis double bonds create a kink. So the fatty acid tail comes down and then it bends out. It has a kink in it. And because of these kinks, that makes the fat liquid at room temperature. Now, you think of like corn oil, olive oil. So plants will produce oils. That's where we get our unsaturated fats, or in our plants. And also fish. Think of fish oil, for instance. Now, of course, we're familiar with trans fats. We hear about them in the news. But what a trans fat is oftentimes what has some companies have done is they say, well, we would like to use this oil and sell the oil, but we'd like to sell it as like a spread. Maybe you can put it on bread or something like that. So we need to solidify it. Well, to do that, what we can do is we can add hydrogen to the double bonds. So there's a commercial process by which they can add hydrogens to make these double bonds go away and then saturate the fatty acid chains that were previously unsaturated. So we call those hydrogenated vegetable oils. And that process of adding hydrogen to get rid of double bonds and to saturate the unsaturated fatty acids, we call that hydrogenation. Now, the thing about trans fats is that they competitively inhibit, and we'll talk about competitive inhibition later, they competitively inhibit essential fatty acids from being metabolized, like omega-3s. So that's the real downside to these trans fats, is that we're not metabolizing some of the essential fatty acids like we should be, and so that's not as healthy. Now, another type of lipid. So we've looked at fats, saturated and unsaturated. Now we're going to look at another type of lipid. Look at this lipid. In this lipid, we still have the glycerol. Here's one carbon of the glycerol. Here's the second carbon. Here's the third carbon. Notice what's coming off the third carbon, though. It's not a fatty acid tail. Instead, it's a phosphate with some other atoms attached. And this phosphate, along with its attached atoms, have charge. That's important, because that charge, the phosphate and these atoms up here, you can see the oxygen has a negative. This nitrogen has a positive. That makes this part of this molecule hydrophilic, like water. Okay? Now, the hydrocarbon tails are still carbons and hydrogens and hydrophobic. Okay? So, what we're looking at in this diagram, we're looking at a phospholipid, which is a very important member of cellular membranes. Okay? So, phospholipids have two fatty acids attached to a glycerol, and that makes up the hydrophobic component. And then, of course, we pointed out the negatively charged phosphate that's attached to the third carbon of the glycerol, and that's the hydrophilic part. And I've already said that phospholipids are a critical component of cellular membranes. Now, when we talk about cellular membranes, and we'll spend quite a bit of time on them later, but here's this gray part here. We'll call this the hydrophilic head, or we'll call it the phosphate head, phosphate-containing head. That's what this gray part of the drawing represents, is this part. So inside this gray part is the negatively charged phosphate and that positive charge nitrogen. And that charge loves to interact with water. So this is hydrophilic. These tails are these hydrocarbon tails. They're hydrophobic. Okay? So you have one layer. This is one layer, okay, of phospholipids, and here's the other layer. Now, one of the things we notice is that the heads of a phospholipid will face, say, the extracellular environment, which to a large degree is water, or the cytosol of a cell, inside the cell, which is to a large degree water. And look at the hydrophobic tails of the two layers. They face each other because they're hydrophobic. So oftentimes we'll refer to this as being the hydrophobic component of a membrane, okay, or the hydrophobic zone. So we'll get into that a little bit later. So what we have in our cellular membranes are bilayers of phospholipids. The two layers of tails face one another. The heads of the phospholipid will face the aqueous environments, the cytosol and the extracellular environment. A third type of lipid we want to look at are the four-ringed lipids. We've looked at this when we talked about testosterone and estradiol. These guys are primarily hydrocarbon. And look what we have out here. Lots of hydrocarbon. But look what we have right there. There's a hydroxyl. This O has a partial negative. The H has a partial positive. So that creates a little bit of water solubility, a little bit of hydrophilic component. The rest of it's pretty much hydrophobic. This is actually a drawing of cholesterol. Well, steroids are the third and final category of lipid we're going to look at. And what's common among the steroids will be four fused rings. And what we see once again in the picture on this page is cholesterol, which is a common part of cellular membranes of animal cells. Oftentimes, cholesterol can also be used in the making of other steroids, such as sex hormones. So, this is our third and final category of lipids. Now what we're going to do is we're going to look at proteins. Proteins are really the workers, the tools, and structures of cells. There are literally thousands of different proteins in our bodies doing lots of different jobs. They're specific for what they will do. Now, proteins are made from polypeptides. Remember, poly means many monomers, and the monomer, of course, is the amino acid. Many amino acids hooked together. Now, so the amino acids are the monomers. Now, when we look at the amino acid, look what functional groups we have. We have an amino group. We have a carboxyl group. And those two functional groups are attached to a centralized carbon. We'll call that the alpha carbon. Also coming off the alpha carbon of amino acid will be a hydrogen. And the final member of the amino acid will be what's called an R group. This R group is the variable group. This is what is different for each of the different amino acids is the R group. Everything else is the same. So there are 20 different amino acids. They all have an alpha carbon. They all have an amino group. They all have a carboxyl group, a hydrogen. Where they differ is in the R group, sometimes referred to as a side chain. Now here's the 20 different amino acids listed together and drawn out for you, and the R groups are highlighted in yellow. Okay. Once again, here's your amino group. Now notice it's an NH3+. That's because it's showing it in the ionized form where it's already taken up a hydrogen ion. We talked about that earlier. But that amino group is attached to the alpha carbon. Here's your carboxyl group here attached to the alpha carbon. Now, it's ionized, though. It's already donated as hydrogen ion. Here's the hydrogen coming off the alpha carbon, and then here's, in this case, the R group, which is, in this case, only a hydrogen. This R group's a CH3. This is a more complicated R group. But all of these R groups, if you notice at their tips, it's either a hydrogen or it's a combination of carbons and hydrogens at the tips. Well, that makes these amino acids here non-polar amino acids, or they have non-polar side chains, non-polar R groups. And so those R groups are hydrophobic. If we look down here, what we find at the tips of the R groups are either OHs. Now I know there's a CH, but it doesn't matter. There's still an OH, SH, OH, some combination of NH at the tip. Anytime you see OHs, SHs, or NHs at the tips, what you're looking at are polar side chains, polar R groups, and they're going to be hydrophilic because the bond between an OH, SH, or these NHs will be polar covalent bonds. And so the N's, the S's, and the O's will have partial negative charges, and the H's will have partial positives. And then lastly, what we have, we also have some amino acids that are electrically charged, some of which are acidic, which means they donate protons. And look, it's going to be a carboxyl at the tip of each of the acidic electrically charged amino acids. Some electrically charged amino acids are basic, which means they take up protons. And so there will always be like an NH, some combination NH with a plus. Here we see NH3 plus, here we see NH2 plus, here we have NH plus. But that means the N is taking up a proton. That's why there's a plus symbol there. And so since they take up a proton, once again, they're basic. So amino acids are divided into nonpolar, polar, electrically charged, acidic, basic. Now here we see two amino acids being joined together. This amino acid is being joined with this one. And notice it's what? The interaction of a carboxyl group of one amino acid and the NH2 of the second amino acid. It's going to be a dehydration reaction, so we have a hydroxyl coming off the carboxyl. We have, or I should say an OH coming off the carboxyl. And we have a hydrogen coming off the NH2 group. And they're put together by way of an enzyme in the dehydration reaction. That way, there's going to be a bond between this nitrogen and that carbon. And that's what we see right there, that nitrogen, that carbon. So I'm going to draw a line here, imaginary line here. And to the right of that line is one amino acid. To the left is the other amino acid that we were looking at here. So amino acids are joined together by way of dehydration reactions within cells. And this can lead to the production of a polypeptide or protein of a specific shape that can do a specific job. So once again, the function of a protein is certainly dependent upon its shape. Now, what really determines the shape of a protein? And the answer to that is primary structure. Well, what do we mean by primary structure? Well, here we have a protein or a polypeptide, and I'm going to give you its primary structure. All right, here we go. The primary structure of this polypeptide or protein is this. The number one amino acid is valine. And then we have a second amino acid. I could give you its name, and the third, and serene's the fourth, and lysine the fifth, and so on, right? So if I give you the name, the sequence of the names of the amino acids, I'm giving you the primary structure. So the primary structure of a protein is the sequence of amino acids within the polypeptide. Now, something else, though, that plays a role in the shape of a protein is the secondary structure. And that's where you have hydrogen bonding going on between different amino acids to create kind of like a spiral staircase look. We call that an alpha helix. Or kind of a folding like you have with an accordion. It's going to be hydrogen bonding again between neighboring, or I should say, between amino acids, different amino acids. Those are called beta pleated sheets. So secondary structure is created by hydrogen bonds that create spiraling or folding like you have in. Now, there's also a third structural level, and this is the tertiary structure. Now, tertiary structure is where you have some R groups that like each other, and so they form maybe an ionic bond. There's an ionic bond that can form between R groups. There can be, hydrogen bonds that form between R groups and so on. So tertiary structure is what we have here, where it's really the overall kind of the three-dimensional shape of a polypeptide due to R group interactions. Now, if a protein consists of more than one polypeptide, there can be a quaternary structure. And that's basically where all the polypeptides, whether it's 2, 3, or 4, that have a tertiary structure, combine in a way and give an overall fourth level structure. That's quaternary structure. Now, the two quaternary structures are fibrous and globular. Fibrous being like a fiber, Globular, of course, I think that name pretty much tells you what it looks like, kind of a globular protein essentially. Now, some functions of proteins, we've said that proteins are the workers, the tools, and the structures of cells. Well, some of their functions include being enzymes. Now, enzymes are responsible for speeding up chemical reactions. So they are really important in living things because, once again, living things are at the very least bags of chemical reactions. And this is probably the most important type of protein there is, the enzymatic ones. But that's not to discount the others. Transport proteins are really important as well. They can move all kinds of materials in and out of cells. We have defensive proteins that are really important to us. Our antibodies, they help defend us against potential pathogens or toxins that can do us harm. We have signaling proteins such as insulin that helps regulate blood glucose levels, very important to us. We have receptors embedded in our plasma membranes of our cells that can receive chemical signal molecules, and then the cell can respond, can change its behavior. They're important. Our muscles are made of contractile proteins for getting us around. We have structural proteins like collagen so that structure is maintained in tissues and things. And then, of course, we even have storage proteins. So that way we have a source of amino acids, a supply of amino acids for when we need to build proteins. Well, the final member of biologically important molecules are the nucleic acids. Now, what we have in the drawing down here below is we have a nucleotide being shown, and we have all three parts being shown. We have a nitrogenous base, and in this case, they're showing the A nitrogenous base. Remember we said there are A's, T's, G's, and C's in DNA nucleotides? Well, we're looking at the A one here. But guys, the nitrogenous base will be attached to a sugar, and then the sugar will be attached to a phosphate. So those are the three parts of a nucleotide. Phosphate attached to a sugar. Now in DNA, the sugar is going to be deoxyribose. Remember, os is sugar, and deoxy, you should look at oxy and think of the word oxygen, and D means away from. It has one less oxygen than the other, its cousin sugar called ribose. Ribose is the sugar that will be used in RNA nucleotides, and we'll look at these in more detail later. Now, we've already mentioned the different kinds of nitrogenous bases. Here we have drawn up adenine, but there's also guanine, cytosine, and thymine in DNA nucleotides, and we represent them with A, G, C, and T. But in RNA, we have adenine like we did in DNA, we have guanine like we did in DNA nucleotide, and cytosine, but we don't have thymine. Instead, we have uracil. So we have an A, G, C, and U sequence. Now, DNA and RNA are the two types of nucleic acids. DNA, which stands for deoxyribonucleic acid, is the stuff of our genes. It carries the instructions for making a clone of us. So in our DNA, what we do oftentimes is within our DNA, we'll have a gene, we'll make a copy of that gene. But look what the copy form of DNA will be. It will be an RNA form. Once again, you will not find U's in the RNA. I'm sorry, you will not find T's in the RNA, but rather you'll find U's instead. Here's what we do. We make a copy of our gene, which is DNA, and the copy form is RNA. And then we use that RNA copy form so we can then hook amino acids together in a specific sequence. Remember, that's primary structure. And once we get the right sequence, then it'll take on the right shape and it can go to work for us. So RNA plays a vital role. Oftentimes it will be a copy of DNA, and we use different copies, different RNA copies in different ways, and we'll get into those a little bit later. Now our DNA is oftentimes referred to as a double helix. That's because it's made of two strands wound around one another like a spiral staircase. Here's one strand here. We can follow it down. That's one strand with its attached, nitrogenous bases, right? Goes all the way here. But then here's what? Here's the other strand, and they're wound around one another. Okay? That's why we oftentimes refer to DNA as being a double helix. It's made of two complementary strands. Now, what do we mean by complementary? Well, on one strand, let's look at this strand. We have a G. What do we have across the way? A C. So G and C will always be across from one another on the DNA strand. A and T will always be across from one another of the two strands. So A on one strand directly across on the other strand will be a T. Okay? So A always pairs with T, G always pairs with C, and what holds the two strands together? Hydrogen bonds. Weak hydrogen bonds. Very important hydrogen bonds, though. So A always pairs with T, G always pairs with C, and it's these ATG sequences that actually make up our genes.